Hello and welcome to Only Sai No Fi. Don't forget to like, comment, share, and subscribe. So today in this video I'll be talking about vaccines. Vaccines, the most important topic going on right now in and around the world. So let's talk about vaccines. In simple terms, vaccine is something which trains your body's immune system to fight a certain disease before even coming in contact with the pathogen. So that is the basic fundamental idea of a vaccine. It is designed to prevent a disease. That is, now your body has not come across a certain pathogen or infectious agent, and you need to save or train your body such that when that disease or infection arrives in your body or you come across that particular pathogen, your body will be able to cope up with the disease faster. So that is the basic fundamental idea. Next is what exactly a vaccine is made up of. It is just that particular pathogen or an infectious agent which is causing a disease is introduced in the body. Now it's not directly introduced in the body. It is some kind of modified. That is, you can say either it is attenuated, either it is killed, either some part of the pathogen or infectious agent is taken and then reinfused in a certain mixture and then introduced as vaccine. So I'll be talking about what exactly is the mechanism and why it's done. So when a vaccine is introduced, it gives rise to acquired immunity. So as you know, there are two types of immunity: innate and acquired. Innate is basically a non-specific immune response introduced due to macrophages, neutrophils, NK cells, etc. Whatever the cells are, n number of cells which are involved in innate immunity. Whereas acquired immunity, specifically T and B cells, that is when they are very specific to that pathogen, that is when the pathogen enters, then they are activated. So vaccine provides acquired immunity to an infectious disease. Now, this concept of vaccine was introduced by Edward Jenner, wherein what he had done is he inoculated skin lesions caused by vaccinia into the healthy human body. So, what he does was he took this lesions, skin lesions, and he inoculated into healthy humans, which later protected them from this particular disease. Of course, now it is done in a very systematic way. So, yeah, this is the basic principle behind a vaccine. First, when you speak about any infection or exposure to antigen which is capable of causing a disease, this is the antibody titer on the y-axis, and x-axis is a temporal scale, wherein this is the first exposure of the pathogen. Or here it is mentioned as antigen. You can relate it to a particular pathogen. So when our body is exposed to certain pathogen or antigen, immune responses develop. That is adaptive response. This is because when the antigen or pathogen is first exposed, then it is later processed by non-specific immunity. Then specific T and B cells are activated, and then it takes some time for the antibody response to develop. This is called as primary response. When same antigen or pathogen comes across the body the second time. Now, this is the second time wherein there is a second exposure of the same antigen. The relative timescale as well as the antibody titer is higher, so that we can cope up with the disease much faster than we actually initially were without taking the vaccine. So this is how a vaccine works. Next is we'll be talking about how some vaccines require booster doses. That is, first administration of vaccine is termed as primary vaccination, whereas subsequent administrations aimed at inducing long lasting response called as booster doses. This is nothing but induction of memory cells, that is T and B cells. So next time you come across a pathogen, the T and B cells remember it. That is, there is a concept of memory cells. I'll be talking about those memory cells in my next video. So there is a concept of memory cells wherein these cells remember that that our body has come across this kind of pathogen or some epitopes of this pathogen initially, and now we need to develop a secondary response that is faster and with higher capacity. And so, as I mentioned, the neutralizing antibodies in the sera determines the success of this vaccine. Now, there are types of vaccines. These are the type of vaccine wherein first type of vaccine is inactivated vaccine, wherein it is a killed version of the microbe. That is, the bacteria or virus is totally killed and then introduced directly into the body. Next is live attenuated vaccine, wherein the virus or the bacteria is live. Mind you, this is live, but the virulence is lost. That is, it is weakened or it is not capable of causing an infection. Next is subunit vaccine, when only a part of the vaccine, certain sugar or protein molecules from the bacteria or viruses, are taken and used as vaccine. Toxoid vaccines are important in case of bacteria or agents which release toxins such as diphtheria toxin, cholera toxin. So in these cases, toxoid vaccines are used. Next is viral vector vaccines, mRNA vaccines and DNA vaccines. Now these type of vaccines are newer generation of vaccines wherein gene encoding for the particular epitope of this particular virus or bacteria is introduced into the host so that there is a constant supply of antigens, because this DNA is into the host genome wherein antigens are constantly expressed. mRNA vaccines similar to DNA vaccines wherein mRNAs are used. Next is what exactly decides whether the vaccine is idle or can be used. So there are various parameters wherein first is the safety of vaccine, wherein obviously there are four phases of vaccine preparation. I'll be talking about them in the next videos. So they test the vaccine in a number of host organisms, including animals. So after a potential safety concerns and harmful effects to humans are ruled out, this particular vaccine is regarded as safe. So if there is no toxicity in vitro and in vivo, then human trials are conducted to ensure the safety of the vaccine. Efficacy, wherein how much actually the vaccine is useful, so measured with regard to the percentage reduction of a disease in vaccinated people, and there is a control set wherein non-vaccinated people are used. Next is the cost. Obviously, the cost has to be low. The economical point of view is very important, as not everyone can afford a comparatively expensive vaccine. Next is ease of administration. First of all, vaccines can be administered via four routes, that is oral, intradermal, subcutaneous, and intramuscular, wherein oral being the easiest of them. As you know about polio vaccines, that is OPV is used as oral vaccine because these vaccines are given to newborns, wherein oral administration is easier. Next is stability, wherein high thermal stability is desired. This is because of transportation or storage conditions. Because it's very difficult to maintain the temperature constant or stable every time, and hence, therefore, there is little fluctuation or the vaccine being transported or taken out from the container, this, that. So, therefore, high thermal stability is desired. Multivalency. Now, cultures of two or more strains from the same species or microorganisms are used to immunize against a single disease. Long term immunity, of course, there are booster doses. So, ideally, a vaccine has to work for a longer duration and therefore provide long term immunity to the patient. So this was all about vaccines introduction. Next, in the next coming videos, I'll be talking more in detail about how memory B cells are produced, what types of vaccine, examples of vaccine, and I'll be elaborating more on these types of vaccines. So stay tuned.